visiting us from the Mayo Clinic. Doug received his MD from the University of Utah and then spent fellowship, residency, I don't know all these things, but at Duke University. And he's currently the professor of medicine at the Mayo Clinic. He heads the cardiac electrophysiology, part of the cardiovascular operation at the Mayo Clinic. And Doug has done some fantastic work, by the way, in addition to imaging and clinical work and so on. I've just recently read a paper that came out of his group about conduction in a one-dimensional and the effect of beta-adrenergic tone on conduction via its effect on the sodium channel. Or as a matter of fact, looking at the VDT max and velocity, I think, as a surrogate to the effect on the sodium channel. So there's that kind of work, but what we'll hear about today is more of a translational imaging-related work. And I've heard Doug presenting several times some very impressive work on how you combine and integrate different imaging modalities together with mapping to get a more complete electroanatomical map of the heart and its substrate, and we'll hear him today in the context of atrial arrhythmias, atrial fibrillation. So thank you for coming.
Well, I appreciate very much the invitation to be here. I have to say that I'm a little bit intimidated, though, because whenever I get to interact with Yoram, he is one of my heroes, and he's kind of up here, and I'm kind of down here, and so that's a little bit of a problem for me. And then somebody told me that some of you are engineers. How many of you are engineers? Yeah, see, I've never done much with trains, and so I'm not real sure about that. And the only reason why I'm comfortable at all being here is because of the kind invitation and the hospitality. But most of the people that I've interacted with today have Macs. And so that to me made a substantial difference. And the other thing is, as many of you know, there's kind of a long-standing interaction between Duke and here, you know, in the form of John Bueno and do you call him Jack?
He was John here. He changed his name when he moved.
Okay, so
kept the creditors from catching him.
So, you know, John, Jimmy Cox, and Ralph Damiano, and now that I have something on video, I'm going to tell you some Ralph Damiano stories.
Great.
Not. I think I'm just going to go ahead. I'm kind of a clinician, and when I'm not doing clinical sorts of things, like ablating whatever happens to move, then we do in our translational lab look at 4- and 5-dimensional imaging and exactly how you do that and some of the problems that are created when it's done wrong. And then we have been working with autologous fibroblasts embedded in certain kinds of biopolymer scaffolds to create both blocking and conducting strands. And I don't know if I'm going to get to that, but I know that we've talked about that in the past, and that requires a fair amount of imaging. As we do these scaffolds and create those strands, then they have to be put in very, very specific locations. But I think I'll save that for later. What I will do right now, though, is I'm going to give you some disclosures. These are always of concern. These are advisory boards that I'm on, and because of my position with the Heart Rhythm Society, I have received no personal compensation for these. I just don't, for a variety of different reasons. On the other hand, I do receive research funding from a number of different places that support our cell work and our translational work and our clinical work. Some of you have been involved with us in the Cabana trial, which is intended to decide whether or not atrial fibrillation can be ablated and whether or not that's better or worse than drug therapy and what it all means from the standpoint of mortality. And then we have some intellectual property here. And I mentioned Rich Robb very quickly because he's one of my major collaborators. And so much of the work that you're going to see today is covered by that intellectual property, and some of it's licensed to different companies. So there are some potentially problematic conflicts of interest. You'll decide whether or not they are or not, and I think I'm reasonably comfortable in saying that they're probably not. But let's just look at this.
Bennett! Bennett dinner!
I just want to make this point. Pictures say 1,000 words. Some pictures 10,000 words. And some of them take even more than that. And so we've been interested primarily in imaging to convey that 10,000 words. And I know that you are also, as you do work with the ECGI, I think that some of this might be relevant from that standpoint. And so we're going to go through 4 or 5 different steps of this. One is the imaging of cardiac structures. Now, I know that some of you are also cardiology fellows, and some of you are interested in cardiac electrophysiology from that vantage point, so I've thrown in a couple of things for you as well. We'll start with the fundamental notion that anything that you do is done better if you can see it. And that doesn't take a lot of rocket science to figure that out. This is simply an intracardiac ultrasound image. We're looking across from the right atrium down to the left atrium, and you can see the pulmonary veins. Primarily when we ablate, and then we're isolating pulmonary veins, seeing that better with image. This is looking down to the right inferior pulmonary vein, and again, a fairly good view of that structure where atrial tissue stops, where the vein starts. And if we look really hard, then we get a little bit of an idea of some myocardium that actually goes down into those veins. And I put this in from the standpoint of if one is looking at ventricular tachycardia. There's a short axis image that you can get from going underneath the tricuspid valve and looking back up towards the RV alpha tract and the LV alpha tract. And I like to best demonstrate that by Macarena. So the RV alpha tract that happens to have a couple of cusps going into an aortic root, that happens to go into aortic arch. Same thing on the right side. And what we're doing here is we're simply looking in a transverse view. So if we're ablating a coronary cusp VT, this is the left coronary cusp, this is the right coronary cusp, and the non-coronary cusp is right there. You can see just a little bit of the left main right there. This is when the valve, when you're down looking quite deeply into those cusps, this is when you're looking right at the level of the commissures, and you get a much, much better idea. So this is the view that you use if you're trying to ablate a coronary cusp VT. This is also the view that you would use if you were going to go above the pulmonic valve, which happens to be right there. It's also the view that you use if you're going to do an aortomitral continuity ablation. So I make that point only to say that this is a fairly complicated three-dimensional structure. And if we have an electrical signal that's coming from that kind of a structure, then it's relevant to know where the structure is and have some means of identifying what electrical activity is coming from what little nook or cranny that's coming from. And to do that requires some degree of registration. We'll get to that a little bit in a second, but this is now looking up into the aortic root. If we just torque up just a little bit more. And the right coronary artery is simply right there. This is the right coronary artery. The right coronary cusp, this is the non-coronary cusp, and a catheter that's up there. If we're ablating around, if we're circumferentially ablating around the right coronary artery, because the patient's got a right coronary artery VT, it's nice to know where that is. And you simply can't get there from coronary angiography. And so, again, making the simple point that imaging is everything. Well, we use a three-dimensional intracardiac ultrasound system in some of the work that we do. This actually is a TEE view first, and I'll show you some of it later. This is one of the cryo balloons that's positioned up into the orifice of the right superior pulmonary vein. You get a little bit of an idea in the two-dimensional images, the 2D views. It's about 85, 90 degrees sector in one dimension and 15 degrees in the other. Again, the same sort of thing, giving a fairly good idea, blowing that balloon up and letting it down to specify where that is. Well, if you're simply going after a pulmonary vein, then all you need to do is know what the anatomy is, and then simply stay as far out of the anatomy as you possibly can. If we're looking at some of the work that we do, then we'll do a CT, constructed CT image set in the EP lab before we do the procedure, and then segment out and create a volume or object map of the left atrium. But it gives a pretty good look at the rest of the cardiac structures, and again, doing the Macarena, the RV outflow tract, and the LV outflow tracts. But you can see that there is a fair amount of problems with this. You can do all kinds of surface smoothing algorithms. You can take a variety of different boxes, and when you take those, then you can do first order and second order and third order and fourth order interpolations and smooth that up somewhat. But at the end of the day, whatever you're doing is creating a surrogate. It's not the actual structure. When we get to the point of registration, then that becomes enormously critical, because if you're going to register anything that has anything to do with physiology, then you have to know how to conduct a one-to-one point match registration. And well, I'm going to show you some data from others. This is Johannes Brockman, and what he's done here is simply created that kind of CT image segmentation, and then is displaying it on a two-dimensional fluoroscopic image. So this is more along the line of image fusion. But in positioning this, unless there's some detector, some location detector, you can put an apparent three-dimensional object wherever you want. And that doesn't mean it's there. That doesn't mean that that's where the physiology happens to be. So if you have a merged image, a CT image, and then you're going to superimpose on top of that a registered physiology, then it requires some degree of caution. And I know that this is something that you've thought about a lot. One of the ways that we create the volume is we sometimes will use 2D ultrasound, create a 2D image, and then circumscribe around the surface of the endocardial surface of here of the left atrium, create a flat or planar structure, take a family of those, and then collate those to create a volume. And then once that's done with a variety of different structures, then it gives a fairly good look at pulmonary veins, left atrium, from the posterior aspect of the heart. Well, we use that to ablate. And as we ablate, then we're ablating here around the right pulmonary veins. You'll see that we're ablating inside, back around the left pulmonary veins. With ultrasound, it's nice because it's a real-time image. And if you're using a real-time image, then it doesn't have many of the problems that are created by part-time images or something that you created, a day and a half ago in a different rhythm. So again, around the right superior pulmonary vein. Well, the registration piece. This is using more of a Navx system to create a matched image, where there's a creation of a CT image or MR image, and then there's a creation through smoke and mirrors mostly, based on the motion of a Lasso catheter and some kind of impedance ranging capability. But in terms of merging those, many of you know that you have to do that with some set of fiducial points, fiducial markers. The more fiducial markers you use, the more likely you are to come up with an appropriate match. In this particular case, we're using 3 springs. The springs are collapsed, and that's the registration. If you're using multiple, then the registration stands an even better chance of being appropriate. Was that something I said?
I just broke skin.
Yeah. So Price Saunders did this study, and what he did is he used a couple of different means of fusing 2 images. Now, it's not appropriate to call this registration because we actually have 2 different images. We're going to put those together. We're going to call that image fusion, which is not the same as physiologic registry. And looked at one geometry match approach versus a third geometry match approach, and looked at the clustering of the points and used the distance as some measure of error. And in this particular case, using a GO1, that is a substantial amount of error in terms of matching two data sets. And using multiple points, fiducial points, then there is relatively little error and more points cluster at a point where they are actually quite accurate. So then we put in catheters, and we use a catheter within some volume that we think has real meaning from the standpoint of cardiac anatomy. And as you can tell from what I showed you before, in some of those cases, there's about a 2 centimeter error from where you think you are, the anatomy that you're actually trying to reach, and whatever you're rendering happens to have said. But this is a patient that we ablated. This is a pre-defined, pre-planned CT. This is a common antrum of the left superior pulmonary vein, the left inferior pulmonary vein, and then we're ablating around that orifice up in here is the left atrial appendage. And I don't know what you think, but I think that that looks pretty fantastic. That ability to take those ablation dots and, as it were, fuse them or register them to the CT. The problem with that is the location of this CT has nothing to do whatsoever with cardiac anatomy unless you've done something to create a relationship through some series of fiducial markers. We usually use intracardiac ultrasound to make that mark, or you have to do substantial validation to take whatever structure you happen to have and then position it in an appropriate position. So let me just look at a couple of studies with you on this. So in here, using a FAM approach, you can see with this circular mapping catheter, as it's positioned, it goes outside of the boundaries on multiple occasions. It's a problem with FAM, and it's a problem with Navx. So if you're trying to position the catheter somewhere where you think it's going to do some physiologic good, it turns out that with FAM, or again with Navx, even with velocity type image fusion, it could be 1 centimeter, it can be 2 centimeters, it could be 3 centimeters. Same thing here, we think that we've drawn some pretty lines around the right pulmonary veins and then down across the septum around in a subangular location. This, by the way, is not the way that I usually ablate, but it is the way I ablate in somebody that's got a large left atrium and someone who's got hypertrophic cardiomyopathy, where it takes an incredible amount of ablation to actually be successful. This is from an earlier conversation today. Well, if you merge your image with an electroanatomic data set, and they call it cardo merge, but this is kind of referred to as a registry, then they actually have much better outcomes from the standpoint of eliminating atrial fibrillation than if you don't use a merge. Once again, there's a problem in doing this. It has nothing to do with registration. One of the things that I've learned from Rich Robb and his group is, if you're assigning a one-to-one relationship between a physiologic and a anatomic data set, we can call that registration. Simply displaying two images within the same free space and twisting and turning them so that it looks like they're pretty good, that's not registration. But here, there was a suggestion that if at least you went through the effort from the European registry, that it made a lot of difference. If you looked from Tang's experience on using CT merge versus just standard electroanatomic mapping, if you look at procedure duration, it was less, fluoroscopy time was less, outcome didn't make any difference. And if you look at most of these merged type or velocity studies, unfortunately, there's not a whole lot of difference in terms of outcomes. There's differences in fluoroscopy time, maybe differences in procedure time, unless I let one of my fellows have catheter, in which case things just don't work out. Well, you can certainly register some other electrogram, and in this particular case it's kind of complex and fractionated electrograms, and we presume that this is exactly where the location is, that it's someplace near that near the left pulmonary vein, but in point of fact, it may or may not be there. Now, in terms of a little bit more from Prosh Saunders, then if you look at the navigation, there are different levels of error depending on which geometric or fusion points you happen to use. In some of the cases, there are fairly substantial differences. Anterior left atrial appendage, other locations, there's a lot of difference. If you look at the posterior wall and the pulmonary veins, then there's not as much difference because the pulmonary veins are kind of tethered to the lungs. And so that there's less of an issue there. And if you look at the work of Hugh Calkins and Dong, then there is yet another layer of complexity if you're looking at the physiologic information and looking to see whether or not it's reproducibly there. Is it in the same place over 30 seconds? Is it in the same place over 60 seconds? And what they did is they mapped twice to see if their CFAE activity was stable. And here's a set of CFAE activity. These are complex and fractionated atrial electrograms that are supposed to have some meaning, and non-CFAE activity. And there's a pretty good map convergence between, or concordance between map 1 and map 2. And if you look, not really too bad. But they use a very long observation period. So if you're just going to look at a physiologic event that takes 1 second, and you anticipate that you're going to register that to a structure, you know what the structure is, but the structure may be wrong, and that 1 second may not be enough to give you an idea. If you look at a physiologic event that occurs over the course of 30 seconds, and it's repetitive, and we'll talk about that a little bit later, then it may be substantially better. This is more of the CFAE activity, and these are 3 different studies that looked at ablating atrial fibrillation with and without that kind of CFAE utility as rendered from an electroanatomic mapping system. And in one study, there was about a 30% difference. In another study, not a lot of difference from Dave Wilbur. And in yet another study, maybe a 10% difference. So using physiologic information above and beyond what you would get by an anatomic ablation may or may not be useful, and is the biggest problem that we've got right now in mapping in electrophysiology is demonstrating that the presence of that map, like complex fractionated electrograms, makes a whole lot of difference to what you're doing. In my heart of hearts, I actually think that it does make a difference. It certainly makes a difference in a point source tachycardia. But in something that's chaotic, or something like VF, or something like atrial fibrillation, then it becomes a bit more difficult. So here are some slides that I borrowed from Sanjeev Narayan, and they raise the question about not complex and fractionated electrograms, but they raise the question about rotors or spiral waves. And what he does is he points that there have been some studies in sheep atria, and Pepe Helite has done most of those, and some are direct map canine atria, and some of them have a very nice leading edge, a very nice trailing edge, and a very nice fixed pivot point. And some of this is work that Al Waldo's done. The problem is that some of them also migrate. So the question is can you look at that and detect the migration? And what does the migration do differently over the course of a short period of time compared to what the anatomy does over an equally short period of time? You know, is there a difference between both, and what does that do to your registration? Well, he collected a series of points and looked that there was relatively little information from human atrial fibrillation about rotors. And I took a little bit of umbrage about that from some of the stuff that John had done, that when I look at it, looks like rotors to me. And so then looked to see whether a basket catheter positioned in the left atrium and the right atrium that had 64 electrodes would have sufficient resolution capability to specify moving physiology that one could construe to be a rotor. Well, here is one of his maps. And if you look, there is what he's describing as a rotor with a little bit of migration in it that is used to ablate and stop the tachycardia. This is a little bit longer of migratory pathway. You can see the leading edge and the trailing edge in blue and green, and when that's ablated, then the arrhythmia stops, and then what you're going to see in the right atrium is activation coming from the high right atrium and spreading across to the rest of the heart. Well, I actually am incredibly enthusiastic about this kind of concept. I'm incredibly enthusiastic about the notion that someone could show anatomy and that someone could show some physiologic activity. Now, I have to tell you, I'm not sure that I would call these rotors. I think rotors and spirals and scrolls and that sort of thing, I don't know that he needed to do that. I think that these could be called phenomena. I don't know about the resolution of a basket mapping catheter that has 64 electrodes where some of the splines of those are crowded. But I do know that I'm enormously enthusiastic about this kind of concept and the ECGI concept, that if you've got a physiologic event and you register that to the surface of an apparent anatomic data set, if you can interact with that in some navigational way, I'm enormously enthusiastic about that. And so when I make comments about this, I think it's great work.
Are these paroxysmal AFs or?
So, in this study, about 35 to 40% of them are paroxysmal, and the rest are persistence, and 42% of them have been previously ablated. And so if you look, is this phenomena from a previous ablation? I don't know. The second issue is, where is this vis-a-vis known structures? Could this just be something coming from a pulmonary vein? Again, I want to be very, very clear. I think that I have nothing but admiration for what he's done here. And I actually think it's phenomenal, and it is potentially unbelievable utility. I feel the same way about ECGI. My concern with the theme of this talk is, where's the anatomy? Where's the physiology? What do you do to register one to the other? And with what certainty do you have a match that continues that we then can navigate with or navigate to? So validation of pitfalls, we've talked about this a little bit. This is another paper from Dong, and it's simply showing in different colored markers the distance in using landmark registration or surface registration, the distance between an electroanatomic data set and an actual anatomic structure. And the distance can be simplest between 5 and 25 millimeters. Now, does that matter? Well, if you are going to a point source of tachycardia and you're going to ablate there, then it matters. But as we talked about earlier today, you could take an anatomic data set and you could do a volume rendering and create an object map of the heart that looks like a duck. As long as it stays looking like a duck and there's a one-to-one relationship between whatever physiologic event occurs and some portion of that duck, you're fine. It simply doesn't matter. The question is does that prevail? And this is a FAM rendering using an electroanatomic mapping system. This is using a point-to-point approach, and this is using more of a FAM approach. Here are FAM veins, and what FAM is to specify location. Then there's current injection, and the location of the mapping probe, or the lasso, has multiple electrodes, and it has to do with impedance, with a small charge of current being delivered. If you look at that, like Navx, there's an incredible amount of error. Here's a pulmonary vein, there's a pulmonary vein, and there's another pulmonary vein. they're about twice the size of real pulmonary veins. Same thing here, and you can get a little bit of an idea in a couple of different dimensions. Here you can see that there's a substantial difference, where FAM tells you that the left atrium is, and where electroanatomic mapping tells you that the left atrium is. And so it's just to point out the issue of discrepancies based on whatever imaging capability you have and whatever physiologic approach you're using. And when we did this, we looked with three-dimensional imaging methods. We looked with CartoMerge and looked at Don's results and saw the error was about 1.8 millimeters. Here, using Cartosound, we're about 2 millimeters. If you look, it depends on where you are. If you're looking with 3D CT in atrium versus ventricles, then there's substantially more error than if you're looking with real-time ultrasound, regardless of whether you're in the ventricles or the atrium. And so again, this is just the validation steps that we went through to try to make sense out of ultrasound imaging. Again, there's a fairly substantial difference in the work that we did a couple of years ago with 3D ultrasound, about 2 millimeter error, electroanatomic mapping about 6 millimeters and about 5 using a merged CT. So these issues come up again and again in trying to be very, very clear. So this is a robbery in process. And the police arrive, and they're going to go directly to the location of the robbery. And they're going to go straight in to the bank that's been robbed, and yet the physiologic event is in a totally different location. Okay, it was a stretch. It's the best I could do. So image fusion. So again, working with Rich Robb, this is kind of the way that we fuse images. Again, with multiple axial slices. In one of our animal models, it looks just exactly the way that you would expect that it should look. And then we take those axial images and then stack them. And many of you who do image fusion do the same sort of thing. And if the images are axial, and then we do a coronal or we do a sagittal, then the voxels are such that we can create three-dimensional images that actually look very, very good. When we do that, we also look at endocardial and epicardial surfaces. This is the left ventricle, the right ventricle, RV outflow tract here. And in doing this, depending on how you set it, and again, this is kind of the intensity thresholding we talked about, you can actually get a very good idea from the body surface what the beating heart actually looks like. And then using that, an idea of where activation onset and offset is. This is the left ventricle endocardial surface. The dark blue is the leading edge of the activation sequence, and the lighter blue is the trailing edge of the activation sequence. And in doing that with a simple CT data set, we think that we have a little bit of an idea of where a particular structure is at a particular time and how a wavefront spreads through it. This is another point where we're actually mapping and pacing. Again, epicardial surface, endocardial surface, and the pacing point over there that you saw in red. Please.
Is that based on the earliest activation or earliest motion?
Okay, so that was a registered activation map to the surface. This one, if you look at this one, this is actually early motion. So what we did with this one is this is the CT data set, and then it's Doppler tissue imaging.
Okay.
So with Doppler tissue imaging, then what you're actually seeing here is you're seeing a change, and I'm going to show you some strain in a minute. With the strain, you're actually looking at the mechanical event. Now, it's an important question because these are all the people that really have done this work, and this is out of Rich Robb's collaborative lab. It really, it's critical to know the difference because in the infarcted myocardium, you have no clue as to the time of the actual electrical onset and what's going to happen mechanically. You know, if you knew that and it was consistent, you could deal with that consistent discrepancy and still have a pretty good surrogate. This is another one of the images that we've done where we've fused, and this is a CT image set in a dog. And you can see the RV outflow tract and the right atrial appendage went around. And now the question is could you interact with this? Could you actually display a physiologic event? And we're going to be looking now down into through the aortic root. We have an icon to help us know where we are. We have an ultrasound image that's being fused to a CT image, and you'll also see that we've got a basket mapping catheter there. We're going to flip around and go back, and we're going to look in the other direction. Now, with this, the recordings that we saw that you asked about were actually made from a basket catheter on the inside and a sock electrode on the outside. With these, we're now going back up through into the right or the left atrium and out through one of the pulmonary veins. Now, with this, I've shown you what the registrations look like. But I think the same thing holds true whether you're registering from a body surface data set or whether you're registering from something on the inside. The issue is, how do you map? How do you put those together? If you're really lucky, my laptop just crashed. And this might take just a second to fix. So if I can multitask, if there are questions, then I'm going to let you ask those. If you'll just forgive me just for a second here. And I took that out of it. So this now is a completely fused and registered data set that we're down looking into the left ventricle. And we're going to superimpose on that or register to that a VT data set created from electroanatomic mapping. And we're looking around now. And as we do that here, then you'll see coming from fairly far down into the apex and then coming back out. I actually think I have that in a different slide here in just a second. But I do think you get kind of an idea here with endocardial surface, epicardial surface, and I think you get a little bit of an idea of what the potential error is in creating that CT data set. I'll get to that in just a second. Here we're using a CT set from the pulmonary veins. And so we've got those. We're going to register onto the surface of that, and a little chunk of electroanatomic mapping. More importantly than that, we're going to go in and we're going to fuse an image set, which now is the Doppler tissue imaging that you had asked about earlier. So now there's a Doppler tissue image set coming from, it's a blood flow coming from the right superior pulmonary vein. Now, I need to say something about the error that's introduced with that, is that could be done automatically to the extent that you thresholded that, but there's a little bit of overriding that. And so with that, you can see that there's a couple of millimeters worth of error in that process as well. So this is one now where we've created a data set where we have gone in and looked at a patient that has atrial fibrillation originating from the left superior pulmonary vein. And looking inside of it with the data set, we've registered that, again with kind of a one-to-one approach, coming out of that left superior pulmonary vein, spreading across into the rest of the left atrium. Giving, I think, a fairly robust idea of exactly where that activation was coming from. Now that's looking from the external surface. And with both of those, then we use that to target an ablation. How good we are at that is unclear right now, because we haven't used this in the data set of patients we've used this in about 6 or 7 patients, but not a big enough data set to know that that's going to make a difference. And we have issues when we go to do the registration, and it makes a difference whether you're doing a piecewise registration or whether you're doing a total volume map registration. If you're trying to do a total volume, you can see that there's quite a bit of error in the registration in pulmonary veins. If you do it, instead of doing it globally like that, you do it piecewise, such that you take given structures of interest, then the registration is actually quite robust. This is the business of error that's on the order of 2 or 3 millimeters.
So just back to the ablation, so that particular patient came from left superior pulmonary vein.
Yes.
You just isolated that vein?
No.
When you did the ablation, or you did the whole thing?
No, we did the whole thing. It's the same problem that we had back 15, 14 years ago when we started doing this. If we only did those veins that showed us activation like that, then we had a success rate of about 30%. And so this is one of those cases where the
curious to know
anatomy tells you everything for a pulmonary vein focus. But I think the thing that you're interested in, the thing that we're interested in is specifying other places where the relationship is not quite as good.
Plus you ablate one and then another one could be good.
Yeah. And this is looking at the number of points that it takes in order to reduce the registration error, and you can see that it's someplace between 10 and 20 points to really get down within any margin of interest or any margin of assurance. Again, this is what the curves look like, or the histograms look like. There can be a couple that are off, as in way off. In general, then the error clusters substantially less than that. It's about the order of 1 millimeter. So let's look a little bit at navigation. I had to throw this in. If we're going to be doing navigation, then this is what I call mouse droppings, where we're using some magnetic system to create a line of desired ablation around some anatomic structure that we've gone ahead and registered. Well, that is very interesting, but it doesn't necessarily mean that it's going to work, because you don't have any clue that that's where the anatomy really is. So it's yet another layer of potential error in the process. Same thing here. If we're looking at creating a volume through whatever navigational means, automatically, this is with auto mapping from that magnetic system, creating the volume, creating the separate volumes for each one of the pulmonary veins, and having a fairly good idea of where these actually arise from, and then where the ablation is directed. This is mostly anatomically directed. You can see that the mapping makes no sense whatsoever. And using a different kind of navigation system, then we are looking at the actual catheter on fluoroscopy. We're looking on ultrasound, and we're looking on a map. So we have 2 or 3 different anatomic structures to give us a sense as to exactly where we're shooting for, and whether or not we're going to be effective in doing that. Well, in doing this, it takes a fair amount of work to do the registration, to actually believe that where your catheter tip is, that you're actually in that location. Now, ultrasound, by far and away, gives you the best image for doing that. When we do that, then we have a fair amount of success in creating a circumferential ring, but obviously can have gaps, and you can see what the size of that gap is. This is looking at some work that Fifan Ouyang has done, and you can see that in some places, anterior, for example, and superior, and then there's a fairly good degree of catheter stability. In other places, like anterior to the left pulmonary veins, then it's really not that great. There's a learning curve for completing the process, and nobody's ever published data to say how good is this actually in terms of replicating a real anatomic structure. I'm going to skip that. It does decrease fluoroscopy time. It does decrease procedure time, but in terms of knowing whether it changes the ablative outcome, that's substantially harder to know. A different one, FIFON, again, procedure times, getting substantially better, but it doesn't necessarily mean that the outcome of the procedure has changed appreciably. Another study from Andre and Natali, fluoro times decreased, procedure times not necessarily changed. So if we're going to use some physiologic rendering on top of an anatomic structure, it doesn't necessarily mean that the ablation is going to be more successful. It maybe gives us a starting point. If you look at robotic navigation, if you're just doing ablation alone, maybe not a whole lot of difference. Ablation using drugs in this case may be a little bit better of an approach. So there's a lot to be determined. Let's talk a little bit about tissue imaging, and I'll be done here in just a minute. Here we're using a three-dimensional ultrasound system, so that we're actually looking down in on the mitral valve, and you can see the catheter positioned there right on the annulus. Here we've moved it up across the lateral LA isthmus, and we're going to go right over the top of that ridge into the left pulmonary veins. Well, we think that that's a fairly nice image. And if you're doing something anatomically, then it's enough. But if you have a mitral valve reentrant arrhythmia, then it may take more than that to get into that point. We're using now a 3D ultrasound catheter. You can see us looking down into the left pulmonary veins with the left superior and the left inferior and the coronal between them. Well, that's fairly nice. But what's wrong with all of this is it's invasive. And so if someone can come up with and do the work that you've done to create something that is non-invasive, then that is actually of spectacular potential value. So here's a lesion. And with ultrasound, we can actually see the lesion. This is what it looks like pathologically. This is not one of our patients. In a study that we did, we looked with ultrasound and found here is scar, here is a lesion, there's a lesion, here are lesions, there are gaps, and there are gaps. So I think part and parcel of any of this would be able to have a sense that your lesion length really is 3 centimeters and the gaps identified as 1.8, and we went back and closed 11 of those gaps in patients where we could not do electroanatomic mapping because there was too much scar. Everything was red. So an ideal solution would be if you not only could have a map and register it, is to have some sense of what tissue looks like as you're doing the ablation. And this is what it looks like on MR, and you've done some of this. This is from Dick Feld. Henry Halpern has done substantially more. I took out all of the slides about tissue imaging. Being able to do it with a balloon catheter and a laser and looking to see what the tissue looks like would be also of substantial interest. It's just that when you do this, you can't typically see that. These are kind of like exemplary images, not particularly illustrative images. Well, if you knew where the scar was, and this is from Vivek Reddy, then ablation could be directed into gaps around that scar. The Salt Lake guys have been using scar imaging to predict who it is that's going to be effectively treated and who won't be. And what they've done is they've said that if there's no scar or little scar, then the success rates are very high. If there are a fairly substantial amount of scar, then you just will not try. Well, if you did that beforehand with a non-invasive approach, that would be of substantial interest. Now, here, if you look, this is one of our infarct models where we are infarcting. You can see by strain rate imaging where infarct is, and then the superimposed ultrasound gives us kind of a sense as to where we are actually looking. And so with this, we have used a model where we can in a beat-by-beat fashion, look at contraction. And in looking at that contraction, have an idea where specific surface points are and what they do through their cardiac cycles. Here, we're looking at strain imaging. And if you look, it goes through a variety of different colors. Red is no motion. You're going to see that through every cycle. Going through green, blue, and everything else is going to be in motion. And so we have used this to image the actual scar location. So we've looked at, in this study, we've looked at MR, strain rate imaging, 2D ultrasound, and voltage mapping, and have come up with a semi-quantitative way of taking a look at that and deciding which is best for specifying the location of that anatomic activity. Well, you can see it best with MR. Strain is second best in terms of a fixed red zone. This kind of in answer to the kind of question that you asked before, there's a lot of thresholding there too.
Yeah.
You know, any imaging modality that you use, there's decision making about what point you're going to take and how you're going to use that. Better than this, and voltage mapping by far and away was the worst approach that we could use. And we are using as another dimension, and you asked, so where did the 4 and 5 dimension thing come from? We have an infrared camera that we use to look at tissue temperatures. And you can see as this spreads what the tissue looks like from the standpoint of temperature, getting up into the 90 and subsequently 100 degree, and an idea of what the dynamics are of that image, and the latency that occurs and the increment in tissue temperatures that happen even after you've turned off the heat. And so this is another dimension that could be added to our physiologic and to our anatomic images. Force, contact force is a big issue. The higher the contact force, the larger the lesions are. As we've looked at this in some detail, it makes a huge difference in terms of transmurality of lesions. If you're using less than 10 grams, then it's very questionable. If you're using 10 to 20 grams, then you're much more likely to be transmural. And if you're using greater than 20 grams, then you still are in a reasonably good range. I found as we've done contact force ablation with Endosense and with Biosense, that most of the time when I'm successful, I'm up in the 30 to 40 gram range. Anything down in the 20 to 10 gram range tends to be ineffective. The difference in force also makes a difference in terms of geometry. The more force that's applied, then substantially larger volumes are created. And we've done that in RA tissue, LA tissue, RV tissue, LV tissue, and using the response to force as a means of telling us about the elasticity of tissue, and therefore where infarct is and where infarct isn't, is yet an additional dimension for that. This is one of Vivek's where the force is actually mapped out onto the surface of the left atrium, giving an idea of where ablations may have been effective and where they're not effective. So I think, I don't know that I did a very good job of timing my talk, but we're through to the end of my hour. I think all I'll say about external beam therapy right now is, so if you have a non-invasive approach to tell you where an arrhythmia is coming from, why chase it with catheters? Why not chase it with a beta source or a gamma source? Well, you can't chase with beta and gamma, largely because they're not sufficiently focused. And if you look at the penetration, then it's not particularly impressive. If you look at proton therapy, if you look at hadron particle therapy, protons or carbon, then the Bragg peaks are substantially sharper. And not only are the Bragg peaks sharper, and I suppose I can just show you that, you know, a Bragg peak may look like that at 10 centimeters with hadron approaches. And if you look at photonic therapy, then entrance doses are fairly high, and so you may get peaks substantially far away from where it is that you're trying to actually ablate. Now, if you look at a pencil beam from a carbon source or from a proton source, you're looking at a beam that's on the order of 1 to 2 millimeters in terms of focus. So one of the things that we're interested in looking at is simply, why not do what my hero does and map from an external surface, providing one could have some sense of assurance that the registration of that and the underlying structure is robust, and then using an external beam. Now, where we are with this right now is we're doing pre-planning. You know, pre-planning is a fairly elaborate process that you have to do to create volumes, to render the volumes, segment them out, understand where surrounding collateral structures happen to be, specifying what delivery might be made, what the portal might be for that delivery, what kind of an aperture you have, and if you have an aperture of a fixed point, can you time the delivery intermittently of that therapy to be at a specific point at a specific time? And how do you do that? So that's another science, and that's, you know, about 20 more slides, and I don't think I'll do that, except to say we think that there are many things that are facilitated by an externally acquired map, a body surface map, and helping identify where those sources are. And again, as we're looking at proton planning, it is substantially more precise than what you'll get with IMRT, you know, with something that's just kind of your standard photon source. So that's what we're looking at. There's been another group that's looked at that, that's done some ablation and has shown that they've been able to isolate pulmonary veins. It's difficult because it's not focused, but we can dream. And so I think I'm going to stop with my dreaming. I think you got kind of an idea of where we would go in the future. And I'll take any questions that anybody has to ask.
Well, thank you, first of all. So the force of all these imaging modalities put together. But so for these studies, for example, how do you determine externally, non-invasively the location of the source, or?
We have to turn to you to do it.
Oh, so you don't do it.
No.
You're
So-
just devising the ablation process.
So the pre-planning that we're doing is anatomically based,
I see.
and it presupposes.
To develop the methodology for focusing on a certain anatomy.
And what we're anticipating is that we're going to be ready to hit something with a pencil beam about the time that you've got an ECGI that says, well, you're fine with pulmonary veins, but what about right or left ventricular apical VT?
In
Room for collaboration.
In WPW some years ago, and this was like 30 years ago, I remember somebody using some sort of imaging to detect the motion to identify WPW pathways, because they felt that, you know, that would be one place where motion might be able to give you some indication of where the activation was starting when you had a quiescent atrium, and then if it came retrograde from the ventricle, it would, cause that motion, and vice versa. It would start the contraction in the ventricle if it was going antegrade. Have you thought about using that for WPW?
So,
Because there would be a point ablation.
The first study that I ever saw on that was done from Mel Scheinman's group, and they used ultrasound and looked at phase shifts, and looked at that to specify. So anything that we can do with a structure that behaves in a reasonable way could be used to guide the therapy. That's the reason why we did the CT where we superimposed the Doppler tissue imaging on top of that, because the Doppler tissue imaging gave us something besides an electrical signal. Now, if you're looking at something that, so here's the problems. If you're looking at something that's at the aortic root, you may be able to see a point source event, but it's going to be moving. And so you have to deal with the motion. In looking at something at the right ventricular apex or something like that, there's a fair amount of motion that's both rotational and translational. If you're looking at something, say, like the SVC-RA junction, where the pericardial reflections have it pretty well tacked down, that may be easier. But, no matter what it is that you do, it is, one of the problems with this is dealing with respiratory motion, dealing with the motion that occurs over the course of the cardiac cycle, translational, rotational motion. But yes, in my dreams, this would not just be an electrophysiologic solution based on some kind of map. You could see it in any number of ways. But as I mentioned to you, if you got an ischemic myocardium or you got a scar, you might be able to see the hinge point, but you would have to take a fairly close look at that. Can you do that with MR? Answer is yes.
So, what is your opinion about any potential, if any, of HIFU? So there are some animal studies showing that you can do completely non-invasively depositing energy.
So about 2 slides in, there's 2 HIFUs. One's a HIFU study